didn't do too bad yesterday. Okay, so what I decided to do, I restructured what I was going to present based on conversations yesterday. And again, same story. Please interrupt, raise your hands, yell. It should be two-way street, okay? So yesterday, there was a question about genome-wide association study. And what I realized that some of you are not familiar with the general methodology of genome-wide association studies and what we're getting out of those. We're not working on any methodological development right now, right? So what I decided to do, I just included all the versions of almost teaching slides. So we'll be introductory remarks about GWAS. I wouldn't talk about linkage methods, so those of you who study classical genetics and know about linkage studies, about mapping using pedigrees, I will not talk about that. So I will talk about three things, and there will be some disconnect. So we'll talk about association studies, we'll talk about random variant sequencing studies, very briefly. Again, if there is an interest in mathematical details, there are questions, I always can jump to the whiteboard. And then we switch gears because a few people were very interested in Mendelian genetics. Trying to figure out whether most of those people are still here. And about new work. So I will switch to Mendelian genetics, but this will be almost a research talk. So that stuff is very new, almost unpublished, so there will be difference between the two parts. So the reason I decided to talk about gene mapping before talking about the function, because we'll be discussing applications of function prediction into genetics, and it will be very hard to do it without understanding of GWAS and without understanding program studies. Okay? Shall we go? Good. So I'll start with gene mapping by association. And the whole idea of gene mapping by association is, the conceptual idea is very simple. It's much simpler than classic linkage, much simpler than many other variants of gene mapping. Right, so the idea is, that if I have, for example, my disease cohort and I have controls, and I'm looking at certain variants, I can detect variants or alleles that are more common in one group versus the other. So in this case, more common in the disease group than in controls. It's as simple as that, right? So if I detect that this allele is more common in the disease group than in controls, then probably this variant is causally implicated in disease, right? So it's causally involved in disease. If my phenotype is quantitative, like height or blood pressure, what I can do, I can run linear regression, and regression is of phenotype on genotype. Now, the classic model is linear dosage, so I regress on dose of risk allele. And you will ask me why there's no dominance, why there are no interactions with other loci, and the truth of the matter, nobody can find any. There is a recent publication out of the Niels lab and the others suggesting that signal for dominance is near zero in genome-wide association studies. Same story. There is, I think, a recent preprint from Surumanian lab at UCLA. So same thing. So they feed pretty complex models, and they cannot find any evidence for additional variance, additional signal due to dominance. So people always pick linear models. So this is all very simple, right? The problem is that the variants are not independent. Imagine that there is an organism which is asexual. So I have bacterial genetics, or I have cancer genetics in somatic side, right? I cannot map anything by association, because I find this variant, and every other variant in the genome will be fully correlated to this variant, right? Because there is no recombination. So in gene mapping by association, the same story as a linkage, recombination provides you scale. Recombination provides you positional information. So when you're learning positional information about the variant. You're testing for presence of the allele. You're not testing for the position. In a classic linkage test, you're testing for the position. You're testing for presence of the allele. But in reality, what you're getting positional information, and recombination history in the population is the mechanism that provides you with information. Okay. I'm looking at your facial expression, and I think it doesn't make any sense what I'm saying. So let's try to do this. So we'll talk about linkages equilibrium. I mentioned it multiple times yesterday. We'll come back and we'll see if it starts making sense. If it doesn't, then we'll stop and discuss, because this is the key point here. So why are variants non-independent? So we discussed coalescent genealogies, and if I see genealogy here, right, I may have multiple variants. So you remember this, right? So time goes backwards, this is my sample, and this is my history of the sample. That's my genealogical tree, or coalescence tree. And these are different mutations in the locus. So these mutations may be in different positions, but in the absence of recombination event, these variants are fully dependent. And this is what we call linkages equilibrium. You can think of linkages equilibrium as just covariance between genotypes along the genome. So for example, historically, in my original population, I was segregating this variant, so there is a large and a small. And at a certain point, I accumulate a new mutation. Right? So for example, in this locus B, I accumulate B small. Now see what's happening, right? So B small is linked to A large. Out of all four possible combinations, I can have A large, B small, A small, B large, and A small, B small. I never observed this one. A small, B small is never observed. So this event, introduction of a new mutation, creates correlation structure in the data. I need a recombination event to break this correlation. So, same story, different slide. I have individual chromosome, like this, so I have a de novo mutation event. And this is my mutation, and this is my marker, or my other variant. And with time going forward, if this variant increases in frequency, recombination starts chopping up this block. So size of this haplotype that is linked to this variant is getting shorter and shorter and shorter, progressively shorter, right? So I'm breaking this correlation down. If I wait for long enough, the correlation will be completely broken. Again, recombination is the force that breaks it. Now we have several measures. So the original measure was suggested by D.K. Watson, who unfortunately passed away last year. This measure was covariance between genotypes. So this is expected frequency of the haplotype. Observed frequency of the haplotype minus expected frequency under the hypothesis of genetic independence. Usually, it's convenient to use measure D prime that normalizes by the greatest possible D under given the frequencies, right? So basically, what D prime measures is whether the haplotypes are completely linked or they're broken. Right? In the case of complete equilibrium is 0, in case of complete linkage is 1. And there is correlation coefficient. Standard correlation coefficient are squared, so I can run correlation coefficient between these two sides. And what happens, just to provide you the intuition, if every combination has 25% on each frequency, right, this is a total equilibrium. I observe all four combinations at the same frequency. The frequencies is 50% and 50% for alleles, and for haplotypes is 21%. My D prime is 0, R squared is 0. Now, if there is shift from the balance, right here there are clearly dependencies. Now I generate D prime and R squared somewhere in the middle range. Here, see what's happening. There is full linkage. I never observed fourth haplotype. But they are not fully balanced, so R squared is not 1, but D prime is 1, because there is no single recombination event here. And here, of course, it's called complete tagging, so this is the problem with associations, right? Because if I measure association on this side, and I measure association on this side, I get identical information. There is no way in the world I can figure out which side is actually causing my phenotype, right, by running the association test. So that's the issue with linkages equilibrium. Now, people frequently draw these pictures of haplotypes. I have the sequence, and I have my risk SNP. For example, my risk SNP is this. Right, so this is where the action is. This is my variant associated with my trait. Now here I have a perfect proxy. So this guy tags this identically. Right, so there's no information gain. There's no statistical method which I can use to tell me that this is actually a functional variant rather than this one. And then there is a tagged SNP. So this variant sits within this haplotype. Right, so it's tagging, and there is enough correlation here that if I find correlation with C, right, I know it would probably be with G here rather than C. Okay? So I'm running a bunch of independent tests, but in reality I'm running dependent tests. And the issue is that if I have very tight correlation structure, I cannot resolve. The mapping. So this is why none of these methods work in asexual systems, right? In bacteria, in somatic variation in cancer, nothing works like this. Gene mapping fundamentally doesn't work. Okay, so if I look at the frequency, change of frequency VLE, yeah, I think I'd like this to sync, so this is why I'm showing the same story from multiple angles, okay? This is change in allele frequency, so I introduce the mutation and it goes up in frequency. And my D prime here, right, I have all of this region length. But even though I have all this region length, the power of association test, so if I would run regression, this is a little bit subtle. It's not difficult to see. I don't think we have time for real derivation, but what matters, correlation matters for confusing my association result. Because this haplotype is totally linked, so I cannot distinguish my mutation from anything else in this block. However, allele frequency is very low, right? So I have no power to detect association. Then it starts being broken, and the region which is completely linked becomes narrow and narrow. However, in terms of R square, my region with high R square actually becomes wider. Because R square also has frequency information in it, and I increase frequency, right? So I increase correlation. If you run correlation and something is very rare, even if perfectly tagging, correlation is still low. Okay, this idea is clear, right? So the idea, like at least non-mathematical version of linkages equilibrium is understandable. So we have history of the population, and that is basically insufficient amount of recommendation for our population history. Now, traditionally, genome-wide association studies, and we're now increasingly moving towards sequencing data. We're in this weird time where most of studies are still genotyping microarray studies. So I'll talk about genotyping microarrays. These are all slides. I see some people disapproving. I would insist that still most of the data we have are genotyping microarray studies, but sequencing very rapidly catches up. Right, and we'll talk about how sequencing catches up. Okay, in terms of the microarray studies. So what's happening is I take my population and I measure frequencies of variants. So I sequence some number of individuals. I know the site is polymorphic, and I can use this cheap technology to go to a much larger population and genotype. So everybody is a heterozygote at the site, or homozygote, or the other homozygote elite, right? So people can be, in this case, GG, TT, or GT genotype, and I can identify this for every person. Now what I'm doing, this is called marker because I don't know whether this variant is involved in my phenotype or it's not involved. What I'm hoping that the causative variant is linked to this, so it's highly correlated, and when I find this association, it points to location of the causative variant. Right? And the only reason it can point to location is because LD extends certain distance and doesn't extend long distances. Okay, so this is the study design. Now, before we go forward, maybe I would, you didn't say anything, but I watched you're nodding and disapproving, so I can give you a quick comment. So what's happening is initially in the study design, LD was a blessing, because if you want to use microarray technology, right? Microarray technology doesn't give you the full all the variation. So you have a chance to find the locus without typing every single SNP. Now with the sequencing data, LD is actually curves, because what we're interested in the functional variant, and we'll talk later today, where we'll talk about functional variants and predicting and finding functional variation. And you cannot find a functional variant, right? Because, again, statistically, all of this group is highly correlated with each other. There is emergence of new statistical techniques that I'll only mention in passing, or again if there are questions, which is called fine mapping. Can I use this correlation structure, carefully model it, to predict which variant actually provides the signal? Okay? Okay, any questions here, or I move to mathematics behind Jumas? I'm going now, nobody else can. I think I'm going. Well, why is it difficult? So it seems that it's a very simple proposition. I just measure regression at every site, right? And then I find my loci. The problem is that there is structure in the data, so it's not only that variants are correlated with each other. Individuals in the population are correlated with each other. Also, we sometimes don't know all genotypes, there is technical variation, there is hidden relatedness, which is another way of saying individuals are correlated with each other. Right, but most problem is here. So the reason this is a problem, so we discussed yesterday that bulk of common genetic variation is shared across population. However, frequencies are not exactly the same. So what's happening is, if I'm looking at one population, I'm trying to detect the signal, right, and my sample is actually mixed. The difference in frequency I will read out would be difference between population groups, rather than difference between individuals with different phenotype, right? So if I'm running a study of diabetes, I can find difference between population groups rather than differences actually leading to diabetes. And that's the key insight here. So this is a correlated data structure. Why is this a problem? And the problem is twofold. So one part of the problem is that if I have some environmental component correlated to ancestry. So the classic example, if I would study fine motor, like maybe not here, but say in Boston, we'd study fine motor skills, and the way we'll do it, we'll ask how well people can use chopsticks when they eat, right? And clearly what we'll find all ancestry component of, all components of East Asian ancestry rather than fine motor skills, right? So that's totally misleading. So this is in the situation where there is slight correlation between the environmental effect on the phenotype and the ancestry effect. And this induces correlation with the frequency, it's total null. That's problem number one. There's problem number two. Problem number two happens when there is a real signal. And the way you think about it, you can think of population structure as linkages equilibrium that is not localized. Right? So in the ideal world, all correlation structure stays within the locus. Right? I have neighboring variants are highly correlated. I move out of the locus, this correlation breaks down. Imagine that I have a sample of individuals of, I don't know, South Asian ancestry and Native American ancestry. And there will be a bunch of frequencies in the sample, right, bunch of alleles that have slightly different frequencies between the two population groups. When I look at the mixed sample, I would realize that I have a correlation, because if I see variant with higher frequency in Native American group in this locus, there is information about possible frequency of another SNP with higher frequency in Native Americans. Right? So I create correlation structure along the genome. I create LD along the genome. It's not localized anymore. I'm losing positional information. So what's happening in this case, I would detect true association, but I would have another signal, maybe on a different chromosome, that reflects the same association, but it's completely not real, right? And I would have multiple of those. So what happens is that I have two correlation structures here to think about. One correlation structure is between variants, and another correlation structure is between individuals. And I have to take into account both of those. Okay, what do we do? So the classic method, I think I removed mathematics. How many people know what principal components are? Many do. So principal. Okay. So the classic method to do it is to use principal components. You can reduce dimensions, right? So you can take your whole group. You can run principal component analysis, and you can plot your samples in principal component analysis. So this is, I think, back in 2008. This is a famous picture by John November and others. How map of Europe. By the way, Turkey is Europe according to the study. I know it's politically from comment, but so you see, Turkey is here, right? So there is Greece and there is Balkans is here, and I don't know, Portugal and Spain. The UK is here, Switzerland, Germany, France. So you basically have reconstruction of map of Europe based on ancestry where people come from, pretty accurately. Meaning that genetics within Europe is determined by geography. It's pretty much determined by geography. Is it true for every single country or every single continent? What do you think? So the counterexample is India. And in India, what happens in India is that there are multiple populations, and due to religious reasons, due to cultural effects, genetic structure of the population is not fully explained by geography, right? So you cannot build the same picture in India. This is the principal components for Indian groups, and they are influenced by, you can map it across language, you can map it across status, you can map it across religion, and so forth. Right? So there are many other possibilities in different populations. So what you do, you use principal components. More recently, people moved to mixed models. How are we doing on time? So there is the ability to use the full covariance structure. I wanted to go real quick through mathematics of linear mixed models. I don't know if people care. Do you care, or you got the main idea about population structure? We just skip it over. Just kidding. Good.
No, said yes, so I said yes.
Okay, so there is a disagreement. Okay, good. If there is disagreement, let's do this. I'll try to go quickly, right? If people would realize how boring it is, you just let me know, and we'll skip anyway. Okay, and if this is exciting, then maybe even people who are disapproving would be happy. Okay, so the general idea of mixed models. Maybe I'll skip this. We'll just do this thing. So look what's happening. So because only half people are interested, I'll try to do it quicker, like showing every other slide. So we have phenotype of individual I, right? So that's Y. And the simplest possible model is that this phenotype is given by non-genetic factors epsilon. That's just what we call environment. I have no idea why it's called environment. It can be whatever, but it's usually called environment. And it's locus effect. So we have J is index of a locus. Beta is the effect of the locus, and we multiply locus effect by genotype. Now, the problem with association studies is that we fit markers individually. Why in the world we fit markers individually? The reason we fit markers individually is because there are 2 million of them. Right? So you cannot run a joint model. So what's happening is we're saying, okay, so this is Y, right? And Y is determined by effect of locus 1, because this is what I'm measuring when I fit individually, right? So I'm trying to estimate by 1. That's what I'm trying to do. And what I'm pretending when I do this test is this all other loci right, behave like the error. They behave like the error too. And that's not right. Why is it not right? So people have said that they know this anyway, so why isn't this right? Because they are not independent. Right? Because different people have different proximity in genetic ancestry. Right? So this is not, this are not independent, independent events. So instead we can fit the model with this fixed effect, so this parameter is assumed to be fixed, and call this a random effect, model all this correlation. So I'm saying that this still is an error term, it's a random term. However, unlike my true noise, which is assumed normally distributed, and independently, this is identity matrix, right? So it's the same, it's a normal distribution for every analysis. U, which is random effect, is multivariate normally distributed, and I keep the covariance, and this covariance is proportional, it's not exact, there should be some scalar here, but it's proportional to genetic relatedness matrix. And genetic relatedness matrix is a covariance matrix over genotypes, right? So that's literally X transposed, and it has the information about covariance structure. So if you have hidden relatedness, if you have population structure in the data, all of this technical stuff, all of this is going into this matrix, and you control for level matrix. Yeah.
Have you ever thought about using an autoregressive model?
So, well, first, it's again, we're not currently developing all this stuff. I'm not sure. I've never seen autoregressive model used in this setting, in this exact setting. I'm not sure it's going to work. We can discuss it maybe offline. So, doesn't seem to be a good idea. Can't tell you right like off the top of my head, but I'm not sure what you will do, what you want to do. Right? Because what you want to do, you want to capture, you want to run regression on a sample with a lot of correlations. Yeah. So what will happen? That's okay. That's an important thing. So what you're suggesting, you're saying that because it's autoregressive, you're saying neighboring locus provides information here. J minus 1, exactly. So what I'm talking now, this is like correlation between loci is linkages equilibrium. I'm talking about correlation between individuals in the study. That's not localized. So you cannot say that. So the distance between neighboring SNPs is not informative. The correlation between neighboring SNPs is not informative about correlation of individuals. Because correlation of individuals depends on every single marker here. Right? So I don't think it's going to work. So this works actually quite well. At least people believe that we're successfully correct for population structure, and there are some bubbling questions sometimes about the data. Okay, so another idea in association study is the idea of a TBAE threshold. So what's important here is, so one thing, by the way, for those of you who are not in human genetics, all of this is frequencies inference, right? So we're just running standard frequencies test. When we're running standard frequency test, the object we're receiving is a p-value. But there are very many tests, right? Because we ran a lot of variables. So this means that I cannot just say use nominal significance threshold. What I have to use is multiple test corrected, right? So what's the idea? The idea is if I'm running 1,000 tests, and if I would use 5% the significance threshold, I will just have 50 bits random. Right, a complete null. I will get 50 signals, right? So that's not working. So I have to correct for how many signals I run. That's a non-trivial question because my variants are correlated, right? So they are not independent. What the community does, and we can question wisdom of the community, okay, here, but this is the standard in the field. So what people do, the standard idea in multiple test correction, the simplest idea is that if I have n tests, right, and my significance level is alpha, I'm writing the probability that out of n tests, I never see it. That's proportionally, that's approximately linear in sample size. So I can do the correction, which is my traditional correction divided by number of tests I run. That's called Booth-Ferroni. And the problem is that the tests are dependent on due to LD. Right? So Booth-Ferroni is a wrong idea. What the community did is it was determined that there is effective number of independent tests, and the human genome. This is very specific for human. Like it's the size of human. It's genetic size of human genetic variation, right? Only common. In rate variation, it's different threshold because this logic doesn't work anymore. So it turns out that the human genome has effectively 1 million common markers. So the accepted threshold, so the standard 5% threshold turns out to be 5 to 10 minus 8. This works. Now the major reason the community believes in this simple idea and nobody's working on more complex methods is because it's working. There is always a debate why we're not Bayesian, why people compute p-values rather than Bayes factors, right? Or like why the world isn't Bayesian, because all statistics is Bayesian, Bayesian is 1, why we're still using frequentist statistics in genetics. It's either inertia or just utility, right? So people understand p-values, people know how to act on p-values, people spend money and time following up the loci. P-value is a very easy guide. It's harder to deal with Bayesian statistics. Now I should tell you that in fine netting, for example, Bayesian statistics becomes becomes quite acceptable. A lot of methods are Bayesian. I will talk later today about localization tests, and again, Bayesian statistics is quite useful. Okay, very classic pictures you read in GWAS tests, right? So it's QQ plot, so quantile plot. You look at expected versus expected p-values versus observed p-values, and usually you want to see something like this. So there is no inflation in your statistics, but there is a real signal deviating from the line, and that's multiple test correction. That's another way to present it. Now the problem with extreme polygenicity is that sometimes deviation happens very early, because we're talking about tens of thousands of signals, and you need better methods to discriminate, and methods like disco regression and others. There are more sophisticated techniques to figure out whether this is inflation in the data or this is a truly very high polygenic signal. And another type of plot you would always see is called Van Houten plot, and this is the p-value threshold. It's 5 10 minus 8, right? So that's called genome-wide significance. Every chromosome, and you're going along the genome, and you see the reason it's called Van Houten plot. It's because you have all of this skyscrapers here. Why do you see multiple variants here at each of these peaks? Why they are not isolated? What is it? Why do I see the shape? So if I see this variant, like I have a bunch of other variants with very similar p-values.
Dependency?
What?
They might have two same dependency on the size.
This is not biology, that's linkages equilibrium. That's literally correlation structure in the data shown in a different way. Right? Because I have local linkages equilibrium, I don't have a resolution, right? If I run individual tests, even if I correct for correlations in the sample, but in this analysis, I don't correct for correlation between SNPs, right? So I don't run a regressive model or something like that. So I always see correlated groups. And this is, so talking to people who believe it's all sequencing data, that's what we're doing most, frankly, at this point. It's very hard to figure out which variants are important. And again, we'll talk with the functional module today, we'll talk about this, because if I tell you that I have 150 kilobases of DNA, and somewhere within this, there is a variant with a minuscule effect size, because I see the significant p-value by running tests of hundreds of thousands of individuals, right? So the effect size is very small. It's very difficult to follow up. So the community is really That's a very hard open problem that we're going to discuss later today, and the reason I decided to swap the lectures is specifically for this. So you would realize how important the problem is. Right? Because I found loci associated with disease. If I go to sequencing data, I have every single variant. I can impute, I truly don't even have to go into sequencing data. I still do not know which of these variants is causative or biologically important. Right? I just fundamentally don't. Okay, so we have about 20 minutes. I spoke yesterday, and again, I apologize, I didn't realize that there will be the same audience for all the lectures, so the one was partially redundant, but what I wanted to do, based on some conversations about UDN project, I wanted to expand this on rare disease, and I would not talk interesting stories about our patients and how individual genes were discovered. I will talk about methodology, and I will talk about what we're doing right now. And I will use our study as an example, but I think it's an example of a broader picture. And at the broader picture, sorry guys, I'm running ahead of myself. I'm running, sorry, forgot everything. I will repeat this again. Now you should realize I'm 7 hours behind you, right? Okay, so one important thing about associations and burden tests, and again we'll talk later today about functional predictions and how this is working. One issue with association tests is that I can only study common variants. Right, so what's happening is that if I have UK biobank size of the study, I have 500,000 individuals, but if I see the variant 3 times, right, there is 0 chance to statistically relate it to anything. Right, 0 chance. I have 5, I'm out of power. So what I can do, however, instead of testing, now this is an important proposition, instead of testing for frequency of each individual variant, I can develop a test that group all variation around some unit. This unit normally is a protein coding gene. People are trying to expand it. We had a paper led by Jerome Link's lab to non-coding windows. We're not sure how well it's going to work. We're now working into having a unit being a network or a pathway or multiple gene unit. But in standard way, it's a gene. And the reason it's a gene, it's very powerful in protein coding regions because we know that there is enrichment in rare variants of larger effects. So that's what we discussed yesterday, right? So this is why protein coding regions is a very good place to look. What's important here is that now I don't have markers, right? As soon as I'm combining variants per gene, they are not correlated by LD, some of them are, but my idea is that I want to look at the association collectively. And I break this notion that genetics is unbiased, right? I start using biology suddenly. Because what I know is that those variants that actually impact function of the gene provides all the signal. And those variants that are neutral or benign, they don't do anything here. Right, so it's very important now to suddenly decide which variants I'm going to combine and how I'm going to combine. And there are many mathematical details how people do it. It's not very sophisticated, but there are a bunch of ways to do it. And again, we'll talk about this particular aspect later today. How does it look? Relatively old story. We were involved in a study on LDL cholesterol. This is the classic Nobel Prize winning paper on the effect of LDL receptor on cholesterol level. And this shows that the effect is pretty sizable. It's 20-fold effect. You never see anything like that in common variation study, right? So you have to go into rare variants. And this is one of the examples in the lipids. This is ApoE5, and I have LDLR, exactly the same gene as identified by traditional familial genetics. And this is how data look like. So this is about 24,000 chromosomes, right? And these are variants collectively in cases and controls. And what you observe here is that all these variants, even these ones, will never be significant on the background of the study. You can never find them alone. But what you clearly see that especially if you take singletons, and especially if you look that some of those are clear loss of function variants, that collectively there are there's increased variation in cases compared to controls. And this is how you find the genes, right? All of them are individually very rare. So that's called burden test or collapsing test. This is the example of LDLR, so this is the same classically identified genes, and these are only loss of function and frameshift variants. And these are cases and these are controls, and you see that the effect is very massive. But to find it by association, you need a very large number of individuals because you need many of these variants to give you statistical power. And if you look at actual LDL level, so I should say that this association is a myocardial infarction. And if I look at the LDL level, again, this is how it looks, right? So this same individuals, again, the association is on the disease status, not on the measure of the biomarker, which is LDL. You see that for disruptive loss of function mutations, LDL is much greater than for important missense mutations, and this LDL level is much greater, so you basically average for synonymous variants, right? So those that are not important for the growing function. And this is why we have to use some idea about the function in construction of the test. And this reproduces the classic paper, the effect size. So that's purely association findings. So you just sequence enough people and running the test, and you're finding the same result. Okay, we'll talk about a little bit more. So there are 3 published studies across multiple phenotypes in the UK Biobank. And why people are interested in this test? People are interested in this test because it provides practical sense, direct road to therapeutic targets. So if you're a pharmaceutical company, right, so the classic work to find target for therapeutics is you're finding a pathway, you have a gene, you're running a whole bunch of experiments, and then you follow these experiments, and then you look at the human genetics, and you're saying, oh, maybe this gene is important. Right? Here you are finding large effect variants with known directionality that work in humans, and you can measure all side effects in humans. So you genetically mimic the effect of the drug, right? You have large effect, you have directional effect, and you know what happens. So there are a bunch of, the first example, I don't know how many of you heard about PCSK9, so this is an amazing gene which is drug target for heart disease now, and there are emerging new targets out of the studies, and that's much easier than working with non-coding genetic associations. So it's a funny situation, right? We're in a situation where common non-coding association explains most of variation in disease risk, but rare variants are of great utility for any practical use of genetics. Okay. Now, yes?
Can we use also this burden test for a diagnostic test?
As a diagnostic test, this is a hard proposition. So, because these are not Mendelian phenotypes, these are complex phenotypes. You're finding the effects. They are large, but they are not fully penetrant variants, right? So you don't know whether the information content is still relatively low. But the major issue is that there are very few. So if you go to this example, right? Look, these are 5,000 individuals. See how many have a variant, right? So you find very small fraction with the variant. If it segregates in the family, then that's a different story, so maybe there's some utility and so forth. There are some people who are saying we should use burden test and identify people at higher risk together with polygenic risk score. That's probably true, but currently none of that is neglected. Okay, any more questions here, or moving to monogenic disease? I'm again running a little bit behind. So you'll see from slide formatting that's a different talk. That's actually a research talk on the subject. And I know there was at least one person involved in UDN, so I wanted to show this work in progress. So what's happening? Again, this is the study I'll use as example. This is bioinformatics methods school, right? So I'm not really presenting one study. I'm presenting the methodology. I'm just using it as the start example. So what's, again, 12 sites throughout the country. We have everybody's genome, and we have currently, so this is the current number of probands. By the way, we always sequence parents. You'll see why, right? So you always need parents. All of this is a story about trio sequencing. Sometimes one, sometimes siblings. We've had historically great success in dominant condition sequencing remote relatives or siblings. So now I'm only talking about sporadic cases and situations where we sequence trio. And the reason we do it because in a large study it's much easier to have standard, right? So to have the same type of data. The idea of the story, as I told you, is that we move rare disease genetics from individual patient, from group of patient, from family, to large data science and large cohorts. And it's surprising how well it works with very heterogeneous cohort, because in this case, again, in order to get into the program, you have to be undiagnosed. Right? You don't have to be neurological phenotype, or you don't have to be, I don't remember if I have, this is the split of phenotypes, right? So it's not study of all immune phenotypes. This is not study of musculoskeletal genetics. This is study of everybody. And still, analyzing data jointly provides a great value, at least in my opinion. Okay, so what we want to do, because we want to find genes that people didn't discover, we want to find signals people didn't discover. So we're going outside of classic single nucleotide variants of indels. In the working group I'm chairing here, it's bioinformatics working group called TBAE conditional loop repeat expansion contraction. I'm not going to present that. And we'd like to find 3 different signals. So one is compelling mutations that arise de novo, recurrent de novo mutations, strong candidate biallelic variants, and also where we're trying to cluster genes by pathways and networks. Okay, so what is a sporadic case? Again, we have healthy parents, nobody in the family shows the phenotype, the child shows the phenotype. One option is that this is a dominant condition and mutation happens de novo. Right, this is why nobody had the phenotype. It's actually 700 per whole genome in the coding regions where all of the action is on average is 1. Which is very good, because if I find a de novo mutation and I have a good suspicion, it's already a pretty good lead. Right? Or it can be a recessive case, right? It can be that one variant is inherited from the mother, one from the father, they happen to be in the same gene. And usually what we see, in overwhelming majority of cases, we see compound heterozygosity. Now, in some cases we see homozygosity. In our experience, at least in our local population in New England, we've had families of Middle Eastern descent, or families from groups like Patan, so they live in the state of Massachusetts, but they remain cultural traditions, or maybe they don't even keep the tradition, but at least in their family history, there are marriages of relatively close relatives, right? First cousins, second cousins, and so forth. Or we had a patient whose grandparents came from 2 villages in Ireland 40 miles apart, and then she didn't know that her parents are related, but if you look at the DNA, there's like 30 megabase block, which is maybe 4 generations, 5 generations, but there was clearly relatedness in my parents, right? So I'm not going to talk about these cases. I'm very tangentially involved in some programs in the Middle East that focus on these exact cases, and I don't think there are any people involved in the audience. I know that in Turkey there is also excess of homozygosity. Okay, so I'm focusing on either de novo or biallelic situation of compound heterozygosity. Now, what would be the method? So we're finding confident de novo variants. We call them with Bayesian method. So this is all the properties I showed the slide yesterday. We have the expected number of single nucleotide changes. We have expected number of indels. All distributions are clean, as expected age with birth. You should run a lot of bioinformatics to call de novo mutations carefully. we wrote a method called novocolor, which is a Bayesian method. Again, I'm not going to talk about that. Made huge difference. All of this, by the way, is very expensive because you run this on the cloud, and if you have to look at original files on the data, that's numbers quickly add up. So these are long and expensive projects. Okay, de novo data. So now we adopted the method published by Sanyor and Rabot University and GenDX. It's called de novo West. And we equipped the method with the mutational model that I described yesterday. So it's much more accurate in terms, well, we hope it's much more accurate in terms of finding true mutations, true de novo events, and correcting for probabilities. So the method works the following way. So we have different functional classes of variance. Again, we'll talk about that later today. We have different measures, so the method relies on estimation of selection coefficient for loss of function. So important selective constraint, we published this in, I think, in 2 papers, 2017, 2019. This metric was adopted by this group. So it looks at how likely the gene is going to cause autosomal dominant condition, and it looks at the functional prediction for the variant. Right? So that's number one. Then we compute expected de novo. That's computed generally by simulations. And there is enrichment score. And enrichment score is run by missense because the idea is that if it's only for missenses, not for nonsenses, and they are enriched in certain region of the protein, this may be gain of function variant. Right? So we have enrichment score and excess score. And when we run this, right, so now again this gives us a p-value, and p-value comes up from combination of 2 p-values and running simulations. This is how p-values, these are known confirmed diagnoses, right? This is fraction of known confirmed diagnoses at low p-values and how much it drops. So you see that there is a lot of enrichment of true signal. And these are our hits. I frankly suspect that a couple of them are probably technical artifacts, which we'll try to follow up. But this is absolutely amazing. Now think what's happening. Consortium of 12 clinical sites has been running for at least 6 years now, maybe 7 years. Every patient was analyzed individually. The fact that in this gene, there are 2 patients having de novo mutation, nobody knew, right? Because you have to run this in others, right? Because it just was never done. Then what's happening is, right, so you see that in many cases the diagnosis is actually matching. But what's interesting is that as soon as you find these individuals, right, in these genes, you find that these 2 individuals and these 2 individuals are very similar clinically. So it's because we keep everybody's clinical notes. So these are new findings, basically new genes for new diseases. Right? The beauty of this is that we never run extensive clinical variation, any biological experiments, right? No, nothing. We just press the button, the statistical method spits out disease genes. Right? What do you have to do? You have to collect tens and hundreds of thousands of individuals. There will be n of million genetics. We'll just find every single gene. Because there are multiple people with mutation in every single gene compatible to life. Right? We'll just find them all. A little bit of stories, right? So these are new findings. So you look at, I know that we have at least a couple people with medical backgrounds, so you can see and decide whether you agree or disagree with our clinical variation. But we pretty much believe it's these individuals have the same, relatively the same phenotype and fit the gene. Okay, this is the story about de novos. Now I have to go quickly because I'm running out of time in my stupid habit here. So again, what's happening here is you can run statistics on this case because in compound heterozygosity you have 2 variants. Right? What is the chance that 2 variants of certain score would land on the same gene? Those are inherited variants. And one important thing here is that we have whole genomes, not exons. So we can also look deep in non-coding region. And we haven't had a lot of success finding regulatory variation, but what we find are variants that produce gain of splice site. And these are not your usual splice variant, they destroy acceptor or donor. This is new, right? Appears in the intronic sequence like Venus coming out of seafoam. Right, so this is literally what's happening. So you see suddenly you have new acceptor or donor gain. What we use SpliceAI, software that's developed by Lumina. It's AI type software that predicts these events. We tested how well it works in collaboration with Rich Sherwood in the experimental studies, massively parallel splice assay. What's good, you don't need patient material, you just put mutations in the measure splicing. This is a quick description of the assay. I think I probably don't have time to go through details here, but that's what's important. So this is how well bioinformatics does versus experimental confirmation. So what you see here, this is machine learning prediction, and this is number of reads supporting acceptor gains and donor gains. And what you see here, there is a pretty good correlation. So we're finding a lot of true signals. The problem, of course, is there are false negatives, right? So we're still missing a bunch of signals here. So it's working great, but not extraordinarily great, right? But this is what we're learning. Okay, anyway, so how it works? It works the following way. So we think that most of the thing that determines distribution and variance among the sequences, mutation rate variation, that as I told you yesterday, we know how to model. So we can compute a probability that out of all mutations inherited from mom, one would land in this gene, in this site with very high prediction score. I can also compute the probability, the conditional probability that score for splicing or score for the coding variant would be above certain value. And I can repeat it from the dead. So I do it once for the mom, repeat it from the dead, and we just take maximum of these two tail probabilities. I've read a bunch of theoretical paper, there was no proven best way to do it, so we took maximum as a simple one works. Again, a couple examples of diagnosis, same story. These are not recurrent, single patients, right? You read about this, and this is clearly true finding. Same thing, we just press the buttons, and all of the diagnostics done purely computationally. The slow process, of course, is validation, right? So clinicians should look at the cases. Sometimes you run additional workup, you may run more organism experiment if you wish, but all the discovery is done purely computationally on a very heterogeneous cohort. So this is, okay, for the research part of my talk, so the main person is Shilpa Kobran. Zach is PI and chair of the department PI of the Cardanic Center. Daniel Traviglio ran all the data, Shamil Lee looked at Shared Genova, Misha Moldovan is a new postdoc, Molly Daniel for mutation rate. Becky, Julian, and Joel are our clinical team, and Rich ran experimental confirmation and the major group of UDN. I think I'm just like 5-10 minutes over, so that's it. So it was a heterogeneous mix of stuff. I hope it was understandable.